Took one.
No, like I got them in order eventually after I figured out I had like 9 things instead of 10.
What?
And then I had 11. And I was like, oh, 11. And I was like, oh, that's extra credit. And I was like,
Yeah, she was all types of crazy.
I was all types of crazy. I don't do well in last things.
Okay, so last
No, she
time,
doesn't.
some of you were here
She's
very late
traumatizing
into the
time for her.
evening, and it was because we had a lot of things that we could do. So if you're thinking about doing some of those other things, like working on your semester projects, which might be a really good idea because we don't have very much time left, then tonight's exercise, this control of microorganisms, you should try to be organized and get it done quickly without a lot of chit chat in between actually doing. So we talked about control of microorganisms outside the body, and that's where we're going to be controlling. And we had a variety of different control measures. So we had physical control, versus chemical control. Our physical control measures, we have temp. We also had pressure and radiation and so forth. I think for this exercise we're using just temperature and radiation, so we're not going to be specifically testing pressure, but we did use pressure to extract chromosomal DNA from bacteria because we beat them with glass beads. So our physical factors are temp and radiation. And our chemical control, we have a variety of different chemicals, but they can be divided into disinfectants, or antiseptics. And both of them are going to control potentially pathogenic organisms, but in different locations. We're outside the body, but if you're using a disinfectant, what kind of surface are you putting it on? You've got a bottle of it right in front of you. What do you do with it?
Disinfect.
You put it on the bench. Is the bench alive?
No.
Not so much. Disinfectants go on non-living surfaces. So there's things happening around you in this laboratory that if you're paying attention, then you can understand answers to things pretty easily. If you're using antiseptics, what kind of surfaces are you putting them on? Living surfaces. So we're going to do this exercise first, and then we also have to do the antimicrobial chemotherapy exercise, which is actually a little bit, it's different. It's by itself because it involves chemicals that are used inside the body. So again, part of this is looking at some things that we've done in the past and paying attention to what kind of things we might have seen, and others have to do with setting up specific exercise activities tonight. So the first thing we're doing is effects of temperature on microbial function. And this is itself is divided into 2 categories. There's some background information here talking about bacteria. We know that we had different categories of bacteria: cycrophiles, mesophiles, thermophiles, hyperthermophiles, etc. And we talked a little bit about why they do that. But exercise A has to do with temperature influencing what's going on inside cells. So our first exercise under A, which is categorized as A1, is effective temp on pigment production. And this is actually an experiment, because what happened, the organism type that we're supposed to use for this exercise, which is Serratia marcescens red, did not grow. So I substituted a different Serratia, and it'll be interesting to see if the effect is the same, because I really don't know if it's going to be. For this part of the exercise, well, okay, so for this whole thing, you're going to be working in groups. I'd like you to be working in groups of 2 or 3 people, so not a group of 6, 2 groups per table, basically. So effective temp, you're going to inoculate 2 plates. you are using Serratia, but whereas we would normally use Serratia marcescens, tonight you're going to be using Serratia plymoutheca. And we want to know if they behave the same way. I don't know. We'll find out. Then when you inoculate these plates, inoculate to get colonies. You know how to do that at this point, I certainly hope. So you inoculate 2 plates, streak to get isolated colonies. You put 1 plate in the drawer. And 1 at 37 degrees. Now, the ones that we're going to put into the incubator, we're going to put them in Ziploc bags. So we'll kind of have to get groups together. We can put 4 plates in a Ziploc bag. The reason I want to put them in the Ziploc bags is because they have to stay in the incubator until next Tuesday. Because if you take them out, then the temperature isn't 37 anymore. The drawer one, you can argue about whose drawer you're putting them in. Although I don't think Serratia plymoutheca smells as bad as Serratia marcescens. Serratia marcescens stinks. Okay, anyway, there's that. So then, 2 is effect of temp on cell viability. And then water. For this one, you have to kind of use your imagination. And imagine that you are a backpacker and that you have been hiking for a considerable period of time. You were hiking in the vicinity of Mount Lassen, and it's summertime. And if you've ever been to Mount Lassen in the summertime, say about mid-August, what you find out is the map that shows you where all this water is, there isn't any of it. Because Mount Lassen area is all volcanic, and in the middle of summer, the streams have all dried up, and so there's no water until you get to a pond. So you get to a pond and it's kind of scummy looking, and you think, can I drink that water? It just so happens that in your backpack you have a Bunsen burner and you have a supply of gas. And so you can treat that pond water and perhaps make it okay to drink. So what you're going to do, you're going to take your pond water. The first thing you want to do is put a sample of this onto a plate. The quantity of cell material that we're going to put on the plate, I think it's 100 microliters. No? 500 microliters, okay. So we're going to put on 500 microliters of water. This first plate you're going to label no treatment. Because you kind of want to know how bad was that pond when you first got there. Then you're going to take your pond water, you're going to put some of it into a tube, fill the tube about 1/3 full. So we're going to be using some sterile, small glass tubes, they're sterile. When you put the water in, fill the tube about 1/3 full. So about there. The cap is about 1/3. So equal in the cap but in the bottom. Anyway, and then you're going to put that into a boiling water bath. So you're going to put it into a beaker, put it in boiling water for 1 minute. Boiling water is about 100 degrees centigrade. If you put it in boiling water for 1 minute, what is that process called? Pasteurization. And then you're going to take a sample of that. You're going to add 500 microliters to a plate. Not the same plate, different plate. And you're going to label that plate pasteurized water. Then you're going to put your tube back in the water for another 4 minutes. learn about boiling. If you boil something, even for as long as maybe 10 minutes, 20 minutes, are you gonna sterilize it? No, because we found out, John Tyndall found out, you couldn't just boil it once. You had to boil it, let it cool for a period of time, then boil it again, let it cool for a period of time, several hours in between boilings, if you wanted to sterilize it. So we can't call the process sterilization. On the other hand, if you boil it for a total of 5 minutes, you've got your 1 minute up here, and then you add another 4, so you're boiling for 5 minutes, it's probably going to be sanitized. You're probably going to kill everything in it that could infect you. So we'll call it that, sanitized. So we're sanitizing our next sample, and we again add 500 microliters to a plate. I think for the media here, let's use nutrient agar. Actually, for this one also, use nutrient agar. So we're going to use nutrient agar for the effective temperature on pigment production, and also for this one. Whoa. If you put 500 microliters on the plate surface, do you think you'll be able to tip the plate and get that to spread over the entire surface? Probably not. So what you're going to need to do is use a glass spreader, so that make sure you move the inoculum over the entire plate surface. 500 microliters is not very much. So you want to spread cells with spreader. For each one of these examples, and make sure that you sterilize your spreader between plates so you don't arbitrarily transfer a bunch of cells. Okay, so that one's fairly straightforward. Now, if you use your spreader, if you sterilize your spreader once, and you spread this plate, and then spread that plate, and then spread the first plate, you don't have to sterilize in between. But if you spread this one first, you have to sterilize before you do this, and if you do this one before that one, you have to sterilize in between. So you should do them backward. The spreading, that is. Okay, then there's some information in here about effects of temperature and pressure on microbial growth. It talks about the autoclave mostly. We talked about that in lecture. There's some information about the effects of osmotic pressure and pH on microbial growth. Anybody remember what our sample was that we had effects of osmotic pressure and pH? They're still sitting back there because we didn't throw them away yet. And we took them out and they were supposed to be sampled. I don't think anybody ever sampled them. Our sauerkraut containers. We put salt in there. The salt created a hypertonic environment, pulled water out of the cabbage, and then the bacteria that were in there produced acid. The pH dropped from around 6.5 down to 4 something. Who did the pH test after a while? Were you guys? Do you remember what it was?
I don't, but I can probably find it.
That's okay. I know it got acidic. Anyway, but they're still back there. Those bacteria are not happy. But they're probably not as bad off as the yeast in the wine containers, because that environment is positively toxic. Okay, so then we come to the last one having to do with physical factors, and that's the effect of radiation, UV light on microbial growth. Let's see. That's B. Or is it C? Oh gosh, it's not even that, it's B. And all of those things have letters. We skipped a bunch. So we're using the effects of ultraviolet light. There's some information about what ultraviolet light is, and then it tells you what you're supposed to do. Well, so again, you're working in groups, and each group is going to inoculate 4 plates. gonna use the same organism type on all 4 plates, but within the lab, we have 6 different organism types that you could use for this exercise. So you can use Staphylococcus or Bacillus, or you could use Pseudomonas or Escherichia coli or Serratia. There's also a possibility of using Proteus for this exercise, but I think your notes say don't. Yeah, don't use Proteus. The reason we don't want to use Proteus is because they will swarm. So where they are on the plate, and then you expose them, after the exposure, they can move around. So they're kind of irritating. Don't use those. Anyway, so you have a choice of which organism type you're going to use. I would like you guys to communicate. I know you can do that because you talk to each other all the time. So when one group decides they're going to use staph, they say, we're using staph. Somebody else say, okay, we won't use staph. We'll use something else. So we have 9 groups. We have 5 different organism types. So it'd be nice if we had 2 groups using same organism type, but not everybody using the same organism type. Anyway, so inoculate these 4 plates, and then you follow the instructions where it tells you what you're supposed to do in terms of exposure. 1 plate is a control. You don't expose at all. Oh, and this inoculation, this is a very different inoculation. So you are not putting these on with a wire loop where you're trying to streak and get isolated colonies. Instead, you're going to inoculate using a cotton swab. By the way, don't flame your cotton swabs. Not between streaks, not at the end. Don't flame cotton swabs. But when you inoculate the plate, this is what you're supposed to do. You have this plate. You're going to use your cotton swab and go back and forth this way. And then you're going to go back and forth this way. And then take your swab and run it around the outside edge. So that you create a lawn. So the idea is to cover the entire plate surface. Don't make this a checkerboard. You know, really make your cotton swab lines close together. So then you have 1 plate that's got no exposure, and then you have a second plate that is exposed for 1 minute with the lid on. Now, this exposure has to be the plate sitting on the bench surface overside down. We're going to expose to a UV light that's in this box, and the UV light isn't on if it isn't plugged in. When it is plugged in, don't be getting down here and look up at the UV light, because it's not good for your eyes. When you put your plate in here, be conscientious of other people's plates that might be in there so you don't mess up their test that they're doing. So you put the plate in, try to put it in the middle so it's directly under the UV light, and expose for 1 minute, lid on. Then you have a third plate that is going to be exposed for 1 minute, lid off. Still in the same position, auger side down, you just take the lid off. And again, direct exposure under the UV. And then the 4th plate is a 5-minute exposure. And I think the lab manual says 1/2 of the plate is covered
One of those days.
with a 3x5 card.
One of those days.
Somewhere along the line, people in this lab decided they didn't want to have just half the plate covered by a 3x5 card. Instead, they wanted to get creative. So we have heart shape, and star shape, and maybe a cat. Sort of a Mickey Mouse. Looks like somebody tore that up.
I think so.
Look, here's a curve. Oh, yeah. There's so many weird things on here. There's this one.
TJ smokes
Okay, so if you just want to cover half the plate, you can use a whole 3x card. So this is the part where if a plate is in here with the 3x5 card on it, be careful that when you put your plate in, you don't knock somebody else's 3x5 card off. It's only a 5-minute exposure. Be patient. Okay. Now for this one, because you're using different organism types, they don't necessarily all go on the same medium type. So I'm not sure where to put this because it's going to get covered up. But for the UVs. And also the chemical thing. Which I'm gonna explain next. For the UV and for the chemical test, if you're using staph, E. coli, Pseudomonas, Proteus. As I said, you're not supposed to use Proteus for the UV, but you can use it for the chemical test. If you're using these guys, put them on TSA. And if you're using Serratia or Bacillus, use NA. Okay, now the chemical test, so this is the UV thing. The chemical test is the effective chemicals, which is E on page 257. In lecture, I talked about different categories of chemicals. So we had halogens, and we had alcohols, and we had heavy metals. And so we have a variety of different chemicals that you can use, and you can see that they're listed kind of toward the bottom of page 257. The iodine is our halogen. You have a choice of alcohols, either ethanol or isopropanol. Our heavy metals are copper sulfate or silver nitrate. I believe what's in the tray is copper sulfate. We do have silver nitrate, but it's a little bit hazardous to work with because if you get it on your skin or on your clothing, it'll make a dark spot, and it doesn't bond very well. I believe it's still in here, though, if anybody wants to use it. We're using our laboratory disinfectant for the surfactant, and for our phenol derivative, we're using carbofusion. Remember, carbofusion is a very potent stain. Be careful what you do with it. Well, okay, so how do we put these chemicals on the plate? You're going to, once again, inoculate a plate so that you have a long culture. And communicate so that not all the groups are using the same organism type. For the effective chemicals, you can use Proteus. But, so now you have all the choices, Staph, E. coli, Pseudomonas, Proteus, Serratia, or Bacillus. You're going to make your long culture, and then we're going to put the chemicals on filter paper discs. So draw yourself a map that shows where you're going to place these discs on the plate. You can also label the lid, but remember, or excuse me, label the bottom. You can label the bottom of the plate so that you know which chemical goes where. But we're going to put the chemicals on filter paper. So you're going to put them on filter paper discs, and I think those are also in your tray. Yes, in that carefully wrapped package. So be careful of these Petri plates, please. These are glass Petri plates. They break. If you have sterile filter paper discs in here, what you want to do is put these on a piece of paper towel and then put chemical on them so that they're saturated with the chemical. Then pick them up with sterile forceps and place them on the plate. There's a drawback to this, and that is paper towels aren't sterile. But I don't think you're going to pick up enough microbes to make a difference. I don't know why those are wrapped in aluminum foil. Anyway, so put the chemicals on the paper discs, and then put the discs on the plate. Put the organisms on first. You're not going to want to try to inoculate the plate around the paper. That's not how you do it. So establish the long culture first. First get your bacteria on there, and then put your chemicals on the filter paper, and then put the paper on the plate. Now, this is Thursday, so these plates are supposed to be incubated, but not over the entire weekend. So if you put them in the incubator tonight, I can take them out tomorrow, and that'll probably work. Everything goes into the incubator except the Serratia marcescens plate that is supposed to stay in the drawer, which is really Serratia plaglutica. Okay, so we'll do this part, and then I'll explain the use of chemicals inside the body, or effective chemicals. That's the antimicrobial sensitivity test, or antimicrobial susceptibility test. I changed the name here. Okay, so I'll pass the organisms out. These, each table gets a set of organisms.
Wants to leave this time, so I'm not doing it. 20 wants to leave, so I'm not doing it. I get too stressed out. I just need to be told what to do.
So we're up to.
And then I'll do
He
it.
was like, let's each do one. I was like, that's.
They're short.
We're going to be getting our stuff right now.
You have the organism types that are on the bench surface, so you have everything to do the Serratia marcescens business. You get to do your effects of UV radiation in your chemical business. I need to go get the pond water, or you can use that pond water to set them.
Oh, that's for the UV, but we're gonna. Yeah.
The pond water.
Yeah, we need like 3 for the first.
Yeah, but I think those were the ones that are being, you inoculate those and then you put them under.
Oh, so it's makes sense. But okay, makes sense. Me, Sunny, and DJ took them all, so. Yeah, there's one left. And there's none left in the fridge. I think she is.
Do we need to get one of those?
Okay, I'm gonna go outside and get some more pond water.
Or are we sharing it with the bench?
Oh, you have a beer. You gotta go see.
So we do. So the first 2. What?
I just looked at her and I was like, we need to boil water.
Somewhat?
I knew we needed to do that, I just didn't know if we could do it first right now.
Never mind. It's not even.
That, you're good.
Now you got a beer.
It's just that thing's really sharp, so I'm not trying to twist it fast. It's jugging in my hand. It's not.
It's lit under here. Oh, wow.
There it goes.
Okay, Rosie. Why would you look at how she's lit?
Well, look at her.
I think we need more actually. Look how many plates they have.
Yeah, we need a bunch of them.
Do we have the big Sharpies? Do any of you guys have the big Sharpies?
I have my small one.
Do they work?
What? How many TSA plates do we need? Well, we need 10.
Ooh, fire alarm. Fire alarm.
I want you to be careful.
Still get more.
We don't have any more. There's not any more
Okay.
nutrient.
No more nutrient auger?
No. Where'd ours go?
Rosie's labeling.
No, there's none up there. It's just TSA so
Yeah,
far.
we need more. We need more, but she doesn't have any.
Not right now.
Yeah, 2 nutrient augers.
She's grabbing more
for
pond
Serratia
water.
marcescens.
So I spread from the back.
Nutrient auger and Serratia. But just say Serratia because that's all she put on there. So I don't know. She didn't tell me about it.
Well, incubator, one in the door. You just reach one of our Yeah, she said we're using like a different restoration.
It's whatever, whoever claims what.
Is it the 16th or the 17th? The 16th.
So when we
You gotta do it on me. It's all about
Oh, okay. So she ran out of plates.
that illustration one that was
Okay, so we'll play over the other ones until
You guys know which one you're gonna do.
right now.
No, not yet. Not yet.
Oh, are you like doing it for the whole class?
Which picture or which one?
She means which one of the things you took.
Oh, but we all have to do serration, right?
No, they're sick. And she wants everybody to try and do something separate so we're not all doing the same one.
I'll come back and speak like another.
Yeah, she said it like 2
So, but we all have to do serration.
No, we don't.
It's on board.
So
No, it's an example.
I think we do all have to do Sarisha and we
And
also do
that's,
the other one.
it's in the lab manual.
I think we're all doing Sarisha plus the other one.
Because that's 1 and 2.
Here's some more pond water. That looks quite drinkable, don't you think? The first part.
Yeah.
So it depends on what part of the exercise you're doing.
I don't know why they wouldn't start with
Alright, so that one's just labeled with the same thing.
They give me the serration. Oh, Gabe has it.
Yeah, I'm gonna do it.
You have the Sarisha one?
No.
So we gotta do 4 of them, right?
Are you done with the Sarisha?
Remember to be very careful in this lab so you're not knocking beakers off of ring stands and spilling boiling water.
It's about a third. Jet.
Each person is going to do a plate with individual participation. You're not doing groups anymore. You're supposed to inoculate 1 plate of Mueller-Hinton auger. You're supposed to use the organism specified for you according to your microscope number. Do you remember your microscope number? Hopefully, yes. There's a list on page 264. You're going to use a cotton swab to create a lawn, and then you're going to put on the antimicrobial discs. We have a handy little dispenser. In terms of incubation, all the plates go in the incubator except the serration plate. The serration will not produce red pigment unless they're maintained at room temperature, so keep your serration plates in the lab drawer. And all the plates are incubated auger side up. The discs will not come off. So pay attention to the chart, make sure you have the right number, make sure you inoculate to get a lawn.
How do I make what number from what?
You're 5.
Yeah, but what does it correlate to?
I think she's gonna bring a tube out.
Just go on the next thing, the way that thing is set up, to kind of see the pattern. On the other hand, I think only two, Stratia and Bacillus have only like 2 feet of these. So we should probably Well, so use either Stratia or Bacillus.
Apparently I'm taking over Aaron's number right now.
Oh, yeah,
My neighbors will come to me.
okay,
I'll go with that.
yeah. Yeah,
So number
yeah, because you guys over there really have like.
What do you need? You're number 5.
So if you move, use the microscope
You guys go hacking outside. Make sure that it didn't get stolen. Yeah, right. Hey, you know.
I think it is.
We're going to do an unboxing.
So your Bacillus, I'm just giving them.
I want to see it here. No. Next week.
Nah,
Nah.
all my stuff's on my bag.
The symbol might be perfect, not be perfect, but whatever. Because when DJ put his in, he turned it, so it wasn't going his. So it might be like half one way, half one. Whatever.
We're doing that.
So get a Mule Hunter auger and do that.
I see your green.
Yeah, don't need to be the straight. Long ones also. Yeah, don't need to be your long.
It was in there. There was another plate sitting right on top. Don't worry about it.
And I swab it with my cotton swab.
Oh, yep, it almost came off. So now we're doing this.
I need this. Oh, yeah.
What microscope number am I?
That's Brian, you're 18.
Guys, what microscope number am I?
What? 6.
6? Okay.
So you need number 3. You are Serratio.
Serratia? Okay.
Is there no?
There was some. There's another. There's another